This is episode 120 of the Immunology Podcast, Empowering Immunity Against Cancer, Dr. Anna Anderson. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rod. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Anna Anderson from Harvard Medical School on the podcast to talk about her research on T-cell response to chronic disease. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
the American Association of Immunologists invites you to submit abstracts of unpublished original research for presentation at Immunology 2026. This dynamic opportunity to showcase your research empowers you to connect, collaborate, advance your career, and access cutting-edge research. Present alongside leading immunologists in Boston, Massachusetts from April 15th to 19th. Visit immunology2026.aai.org to learn more. What's up, Jason?
warble, 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 warble.
Warble, warble. How is that? Do you have your turkey seasoned already?
I think my parents are brining it today, and then they're putting it in the smoker.
Oh, my.
My parents have a new barbecue that smokes, and it's flipping amazing. We've done turkey on it before. It's to die for. And then we're taking it from my parents' house to my in-laws' house because we all live near each other. But my dad had shoulder surgery recently, so I have to go lift the bird in and out.
Oh. Wait, but are you deep frying it or just
No, this is smoking it. It's a barbecue with a lot of smoke involved.
Okay, a bit more healthy.
Yeah. I've never done a deep fried turkey. It's like one of those bucket lists to do once, but you know.
At your age, Jason, I would not consume that.
I am gloriously on Lipitor.
Right. I'm on medication, so I can do this. It's funny because one of my papers is about treatments for atherosclerosis. So, very on point. Very on point. So, this paper is published in Circulation. So I think it's more, aimed at the cardiologists amongst us. But it's called Oxidated LDL Targeted Chimeric Antigen Receptor T Regulatory T cells Reduce Atherosclerotic Plaque Development. And of course, I got it because, they're Tregs. And, I figured, given, I thought, it was a really nice example of how everything is immunology in reality. So atherosclerosis, in a way, is an immunological problem because a big part of the developments of these plaques, I guess is the right pronunciation, it's due to macrophages that, they get activated, they ingest a lot of these lipoproteins and these lipids, and they become foam cells, which are inflammatory. And this generates this kind of local inflammation, the death of the macrophages, and that's how they make these plaques. It's like dead macrophages. They all like kind of put together, and that is a big part of the source of this. And there's a lot of inflammation associated with atherosclerosis. And so basically, what the authors in this paper suggest is, well, if inflammation is the issue, there's a T cell for that, my friend. And that is the starting hypothesis of this paper. So first author, Robert Schwab, from corresponding author Avery Posey at the University of Pennsylvania. These are they know themselves around CAR T cells in general, so that's also what caught my attention. It's a good place to start. So in this publication, they start by looking into, so LDL cholesterol is a lot of that has been shown to be so it's a big part of the development of these plaques, and reducing LDL cholesterol has been shown to slow the progression of atherosclerosis. So basically, that's why there have been a few antibodies have been developed that are specifically bind oxidized LDL, and this seems to help in and there's chronic inflammatory conditions also, psoriasis, rheumatoid arthritis, SLE. These are all also have associated with elevated oxidated LDL levels, and um, that's why some treatments have developed these antibodies or antibody fragments meant to kind of reduce the or capture this oxidized LDL. So you have an antibody. If you have an antibody, you can make a car, and that is what they did. They looked so as part of this project, they generated a few different cars using I think 2 or 3 different antibody that are specific against oxidized LDL, and they tested. These are CD28 uh, CD28 cars, and they basically tested which ones would activate T cells and in the presence of oxidized LDL. And based on this, they chose a construct. So basically, what they show is that they can generate a CAR T cell that is recognizing this oxidized version of LDL, and not the regular, but so not the non-oxidized version, so to say, but the one that is the most problematic. And so from that, they say, well, if we have this CAR T cell, let's put it on Tregs. But as I very well know, and the authors very well know, it's hard to start from Tregs. There are very few Tregs available for you to take, and from in humans at least, to take and to uh, genetically engineer, so to say, to express a CAR in a way that is stable, um, and that will expand sufficiently for clinical purpose. So what they do is they propose a different approach, which is transforming conventional cells into Tregs by forcing expression of FOXP3. And this has been described before as a method of generating what seemed to be fairly stable Tregs, of cells of a Treg phenotype. The forced expression of FOXP3 does result in the acquisition of a lot of the prototypical Treg phenotype in conventional T cells. And so that's kind of the approach that they suggest. So they have this construct, this 2D03 CAR, and they add it together in a vector that also has FOXP3. And they transduce the cells, and basically they show that they can generate cells that have a phenotype that is very similar to natural Tregs, the Tregs that are found in the blood. And they have, the typical markers, CD25 high, CD127 low expression, and that also CTLA4, and also expression of other typical associated markers like GARP upon stimulation. And what is interesting is that so they show in vitro first using the human T cells that they can they do suppression assays and in which they are activated the cells with macrophages that are cultured with are stimulated with LPS, so they uh, will and they add oxidized LDL, and then they by culturing together with these Tregs that are recognizing this oxidized LDL in the macrophages, they can reduce, for example, lipid uptake in the macrophages, and reduce, I think, also expression of some of the inflammatory um, cytokines. So basically, in a way, reducing the activation of macrophages, and they show that to some extent this is mediated by the production of TGF beta by these CAR T cells when they get activated. And so they also have some results on so that is, of course, TGF beta is a well-known influencer of macrophage polarization, and that kind of skews the macrophages away from this pro-inflammatory, quote unquote, M1 macrophage towards a more M2 macrophage. like phenotype, and that is kind of the what they're trying to accomplish with this T-Rex. And then they show that it's safe. They have some, so they do some mouse models. So they have transgenic mice that are expressing the ApoB protein, the human ApoB 100 version of the ApoB protein, which is the one that is present in humans. And basically, it also show that um, by using a murine version of this CAR Trex that is targeting this receptor, they can also um, treat the mice and reduce to some extent the accumulation of the atherosclerotic thing. They actually look into atherosclerotic lesions in mice that are prone to um, to developing atherosclerosis. So I think in general, it's a very interesting concept. I think, T-Rex, directing T-Rex, it's really cool doing with cars, because then you can target this and kind of this unusual non-canonical target. You can basically target anything that's expressed on the surface, and you can just bring the regulatory T cell there, and then it's gonna make, in this case, seems to be TGF beta, seems to be the most important component of these inhibitory program. And I think it's really a really intriguing kind of approach. Um, I can imagine a million issues with this, also because you're having these T-Rex that are based on conventional cells that are going to have a T cell repertoire that I know, would recognize potential, uh, the undesired antigens, and maybe enforce regulate or suppress the immune function against a viral antigen, whatever that is in the repertoire of those T cells, because they're not knocking out the T cell receptor. But I think that as a concept is really cool. And this is something I personally very interested in my own research. So it's like, oh, this is pretty cool. Um, so so if you ever, the statins don't work, Jason, you can just get a nice infusion of those good T-Rex, and they can take care of it. They can keep your immune system at check as well.
It's interesting. I wonder if the cost benefit's going to have to be such that it's for really refractory people with high risk, right? Because CAR T is very expensive. But I could see a case for it, right? Once you're on your second or third heart attack, you're in a bad spot for the healthcare system.
Yeah. Yeah. I mean, to be honest, in this publication, they're not studying the effect of transferring Tregs in a mouse that already has atherosclerosis, right? So this is, haven't gone that far.
Yeah.
So there's a mice that will develop atherosclerosis, and they are at the same time they have the treatment, so they develop the plaques or not. So it's unclear what would be once you already have these lesions pre-existing.
Oh, right. But then if it's for prevention, how are you going to afford that if a CAR T therapy is hundreds of thousands of dollars?
Yeah, exactly. I think it's not really practical. I think as a intellectual exercise is very interesting, and it's an intriguing concept, but I think cell therapies are too expensive to be done in these cases. And I know that very well, because I've been trying to justify my own weird T-Rex, and it's always the same question, like, where do you have to be to justify the expense and the difficulty of these cell therapies?
Yeah.
Yeah, the risk.
That's a toughie.
All right, what you got?
Well, I guess I'll talk about T cells as a segue. So this is a paper it's probably easiest to summarize quickly. It's in Nature. It's tumor reactive heterotypic CD8 T clusters from clinical samples, published the 19th of November. The first author is Sofia Ibanez Malero. Last author is Daniel S. Pieper. So here we go. This gets in Nature because they answered a question with clinical samples. That's very hard to do, and it's very robust, but they found, and I think it's showing that some of our current approaches are missing things, if that makes sense. So we all have taken, we all take cancer cells from tissue and flow the immune cells and look at individual cells and make some conclusions, right? And they said, well, we know there's a spatial relationship between the T cells and the cancer cells. And does that mean anything? But when you do the flow, you usually separate everything out. So they used image flow and did a little bit less of a dissolution prep to allow clusters, and really looked at what happened with heterotypic clusters where there was immune cells and cancer cells next to each other and what the immune cells did. And they had 21 different human melanoma metastases. And they were able to identify that these T cells had a little bit different behavior. So like they use an engineered system to start with as well in mice with specific color markers to be able to track everything a lot easier. And they found that the tumor cells and the T cells form these cell-cell conjugates. Not surprising, but they could show it, right? And that they could replicate this in the clinic. And these cells are really tumor reactive and then they do single cell RNA-seq and map it all through and proteomics. And they show that these cells are effector cells. They sometimes have early exhaustion, early stem cell types, but they're more active and better at killing, especially when injected into other models, into cancer models, than cells not associated with the tumor. So again, I didn't say anything surprising, right? It's not surprising that T cells, CD8 T cells, next to the cancer cells, are better at killing the cancer cells and prime for it. But the point is no one's ever actually, when we do flow before, separated out. It's just a pile of tissue and T cells, and you go to a single stream of single cells, and you don't actually gate on which cells we're touching the cancer cells. And if you do, you realize those are the active ones, essentially. That's what they show here. And I'm basically done. I don't need to, they spend the entire paper showing this. They're looking at antigen presenting cells versus tumor cells, understanding their not so much mechanisms, but the signaling and how that relates to what we know. And they're active. And then they show that it can kill cancer. So really, it's this approach. It's like this aha moment of, guys, we're missing a picture here by just digesting and going. So there you go.
I'm really happy that you cover this paper because it is by my very good friend, Sofia Ibanez Molero. And this Johanna Feldman is a co-first author. And that's author Daniel Paper, not Pieper. I'm not going to give you a hard time about that. But I think it's really, as you said, it's really cool that all this information people were kind of ignoring. You just gate out the doublets, or you just, find all the ways to separate all your cells.
Yeah.
And in fact, there's this very important information that comes from this interaction. And some of the figures are really cool. Like the one, the ones you really see the clusters of cells, and you see what clearly is an immune synapses, or some kind of synapses between the cancer cell and the T cell. And it's there's clearly something going on there. And it's nice that they have really looked into it with so much care. And I'm really happy that this story came out because I've been hearing about it for a long time. All right. So which brings me to my second paper of the day. And today I have, a macrophage. Today the topic is macrophages. I know I'm not very kind to macrophages. I always, just say that they are the trash cans of the immune system and they will eat anything.
Pretty harsh.
I know.
Hurts my feelings.
But they're cool cells. They do a lot, and there's a lot that we can use them for. And this is one of the examples of paper in which they show that we should look at the macrophages with a little more care. We should take advantage of their presence. So the paper is called macrophage targeted immunocytokine leverages myeloid T and NK cell synergy for cancer immunotherapy. Quite a mouthful. Published in Cell. Published the publication that is in the future in December 11th, but it came online on, I think, November 19th. First author is a shared authorship between Michelle from Lockenhien, Pascal Zwicky, and Ken Shi from corresponding author Ido Amit at the Weizmann Institute in Israel. And yeah, I thought it was really cool. It's a really cool story. Basically, um, in this work, they are again, is this in the tumor situation? We know that tumor associated macrophages or TAMs, they they can be quite a problem in immunotherapy, and they can be a major in many cancer types. They are a major part of the tumor microenvironment. They take a lot of space, and they take a lot of they do a lot in the tumor microenvironment, particularly those that are immunosuppressive. And there's one checkpoint uh, known as TRM2, which is key immunosuppressive kind of checkpoint in this tumor associated macrophages. And um, previous work from this group, and I think other parts of the literature, indicate that macrophages that express TRM2, they are really contributing to poor prognosis cancer patients, are protecting the tumor against the immune system. And that is, we need to find a way of overcoming the function of these cells, because even if we activate the T cells, if the macrophages are there, interfering, it's going to be really hard to actually get a proper immune response. So basically, in this paper, they're looking at ways to take advantage of this, of the fact that we know that this marker, or this checkpoint, and the activation of this checkpoint, the triggering of this receptor is important, will increase the immunosuppressive phenotype of macrophages. So we can block this receptor. We can use antagonize, inhibiting antibodies. But what else can we do? And this is kind of what they're looking into in this paper. So they start the paper saying, well, there are, again, antibodies that can block the function of this receptor. And they kind of work, but not a lot. They don't do a lot. We know that they look at some data in spatial atlases of different types of cancers in humans, and they show that indeed these TAMs are often found in close proximity to T cells within the tumor microenvironment, and that is kind of one of um, the most important. So they do all this algorithm, and they have this AI program studying all of these spatial transcriptomics data, and they really come to the conclusion that, and also single cell analysis, that there's this close proximity, physical proximity between TAMs and T cells in tumors. And so there's I think that's where they spark the idea that if you have something that can work both on macrophages and T cells, because they're so close. So they suggest first, well, if you have an antibody that's blocking TRM2, they have a humanized mice that have a humanized version of TRM2, and they show that, well, that kind of doesn't do a lot. Treating the mice with this TRM2 antibodies is not a big, it's not a big game changer. But what they do is say, well, what else can we add to this molecule? So they make this anti-TREM2 antibody, and they fuse it to an IL-2 molecule. They choose a superkine, a synthetic variant of IL-2 that has an increased affinity to the IL-2 receptor beta, which is the one that is mostly expressed on like naive or non-activated T cells, and also, so it's mostly a way of more targeting IL-2 preferentially to effector T cells rather than regulatory T cells that have very high expression of CD25, which is the IL-2 receptor alpha subunit. So there are ways in which you can make IL-2 versions that preferentially bind to the beta and not the alpha receptor, and kind of play with the affinities and the relative levels of stimulation from one subset to the other. And so they do some in vitro analyses, and they show that um, that this molecule works, and then they do an in vivo test in which they treat mice with this what they call IL-2 SK fusion molecule, and basically they give the mice massive toxicity. So they show that although they seem to be able to target these TREM2 macrophages, they the toxicity of this cytokine associated to it is just too much. So they come back and think, well, what can we do? And this is what I think is really cool. They say, well, can we make a version of this molecule in which the IL-2 is only going to be functioning in the right place when it's binding to the macrophages, preferentially in the tumor microenvironment. So they generate a pro-cytokine fusion molecule in which they basically cytokines are fused to blocking domains through a linker that is cleavable by a particular protease. Ideally, one that is expressed in the tissue of choice that you're targeting. So they're lucky that macrophages expressed a lot of proteases on their surface. And so what they do is they make a construct in which they have this IL-2 molecule covered, so bound through a linker, through a protease, sensitive linker to the IL-2 receptor piece of domain. So that under normal circumstances, when they make this molecule, the part that binds to the receptor is hidden by this piece of receptor that is part of the original molecule. But in between, there's the linker that can be degraded by a certain protease. And they test different proteases, and long story short, they choose one that shows really specific activation only after being they have very kind of low baseline activity, and they are very well activated in presence of the macrophages that express this protease, which happens also to be highly expressed specifically on macrophages in the tumor microenvironment. So they're all very convenient, very, very nicely, all kind of fell. It looks like it fell together very, very neatly. I'm sure it was a lot of work. But I think in principle, that's kind of the idea. So they show that this protease, so they have a protease, how they call this a mite. I forgot a myeloid. I forgot where the name comes from. But basically it's a molecule that gets activated by a protease made by the myeloid cells, and therefore only makes the IL-2 accessible in the tumor microenvironment preferentially. And this really reduces the toxicity and still keeping a lot of the functionality of the IL-2 once it's activated. And well, basically they do a bunch of studies. They show that they can have, it works. It has very little systemic toxicity. They can combine it with CTLA-4 treatment, anti-CTLA-4, which is a good way of eliminating regulatory T cells that still kind of get activated to some extent by this new molecule. So if they combine it with CTLA-4, it's a really nice combination therapy that works very well. And they do a bunch of studies, but I think in general, the conclusion is that they can both inhibit TREM2 on the myeloid cells, which then kind of prevents their programming into suppressive cells, or induces a reprogramming into more pro-inflammatory cells with a more monocyte profile that express, more effector molecules and less of the ones associated with M2 macrophages. But on top of that, the IL-2 part also activates lymphocytes. It activates NK cells, activates CD8 compartment. While if you deplete the regulatory T cells through the CD4 antibody, then you have a really nice combination therapy. And yeah, so they have that in mouse models. They also do this patient-derived tumor fragment system in which you take like tumors from resected tumors and you cut them in small pieces of like 1 millimeter by 1 millimeter or 1 millimeter. And then they culture these ones with these molecules, and they also show that they can really show a lot of changes in the expression, the gene expression, and the cytokine expression of this tumor fragment, which is kind of they're like avatars of a human's tumor. And yeah, so they improve activation of NK cells, of T cells, reduce some of the markers of exhaustion, TIM3, LAG3. No, sorry, in humans, they actually don't see that, but basically they see some of the markers are increased expression of perforin, granzymes, um, and some reduction on some of the markers associated with exhaustion. These are very short cultures, so it's not a lot kind of really look at long term, but I think in general they show that there's a shift in the activation of the immune cells in these fragments. So yeah, so basically, I think it's really cool. It's a cool concept, like synthetic molecule, and I think it's very important find a way to not only look at the T cell directly, but also directly target the myeloid compartment, especially in certain tumors. It's kind of crucial for
You can't
success.
ignore it. Macrophages matter.
No, can't ignore them. They're right there,
T cell.
right in the middle.
I'm like, well, it's
Yeah,
a team.
indeed.
All right. So I got one more for you,
Shoot.
which is about fat.
Fat.
So I'm no segue.
Sexy.
It's subcutaneous white adipose tissue derived extracellular vesicles maintains intestinal homeostasis via IgA biosynthesis in aging mice. What? All right. First author is K. Kao Long. Last author is Kenneth King Yip Chang. So white adipose tissue is the most of the fat we have. There's also there's also brown adipose tissue. This is about white adipose tissue, aka what we think about when we think fat, right? And then this is inguinal subcutaneous white fat. So the stuff that's subcutaneous versus visceral, which is important because they control this. We know as you age, fat dysregulates, and we know as you age, your gut is a little more leaky. But we have now found for this paper that at least in mice, they're related. So here we have that in this paper, they show that if you remove the inguinal subcutaneous white adipose tissue, the white fat, you develop a little bit of a leaky gut by zonulin differences in phytidextrin absorption and LPS circulation that matches what you have in an aging intestine. Okay, so it rephenotypes aging. And then you have less IgA in your intestine, which is related to higher levels of segmented filamentous bacteria. When they see that, and they see microbiome shifts, they see that loss of this fat alters intestinal IgA production and population homeostasis. What they don't see is big shifts in cells. What they see is a shift in IgA producing plasma cells. It doesn't alter Peyer's patches. It doesn't alter a lot of function, but it does alter programs in cells that lead them to becoming IgA B cells, right? They then start understanding signaling. B cells themselves require external retinoic acid in other cells to switch. And this is important for IgA class signaling. And they find retinoic acid levels are down in these mice. And so they spend a bunch of time showing that, like, okay, if you reduce the IgA, you have less segmented filamentous bacteria responses, all the things like you see A happen and then B, the downstream effect is real. But that's important. And like the mice don't tolerate C. rodentium infection very well either, all this stuff. But here's what they really show. They show that the adipocytes produce the retinoic acid, excrete it in extracellular vesicles, which is uptake by the plasma cells. It causes them to become IgA producing plasma cells. And they map this whole thing out. They also show that visceral white fat doesn't do this. It's the subcutaneous white fat only. They do other resections of other visceral fat. They can't do it in the peritoneum because it's too risky, but they do it in other locations. They really map out the signaling. They rescue by giving exosomes with retinoic acid. They block retinoic acid production and show the same phenotype. They make extracellular vesicles without retinoic acid and show it doesn't work. And they go down all the rabbit hole. They then used aged mice in the end, and then they rescue aged mice with extracellular vesicles with retinoic acid. And it's damn cool. It's very, very cool. And then that aged mice fat is dysregulated, doesn't produce this, and that's what causes the whole cascade. So white fat produces retinoic acid, which makes your immune cells in your gut make more IgA. That alters your microbiome and leads to a better barrier, and everything goes to crap as you age.
Interesting. Another thing that goes to crap as you age.
I'm just sliding downhill like a bus without brakes.
But then, so retinoic acid from
the fat.
gut, from
it's
the fat.
That's fat. It's synthesized by the fat.
And can you consume retinoic acid to compensate for that loss?
Yeah, but you don't want to just eat it because that's a whole that's like retinol A and all that. Like, that has all types of teratogenic effects in babies. But yes.
Okay.
Retinoic acid inhibitor, I forget. But yeah, there's complications there. But yes, basically. You just have to be careful about it.
Yeah. Well, I guess if you're old enough, you're not gonna care too much about the affecting a potential fetus. So that's
right,
fine.
Exactly.
All right. Well, another way the microbiome is everywhere. You're allowed to gloat.
All right, I am. It's most important. It's there's more of them than us. We have to acknowledge their importance in our lives.
Indeed.
But with that as a point, we're actually about to have a very important, interesting conversation with Dr. Anna Anderson at Harvard in just a moment. But before we get to that, are you looking to improve your T-cell therapy development? With STEMCELL Technologies' new GMP manufactured Immunocolt XF, you can achieve robust T-cell expansion with high cell yield and viability without the use of serum. To learn more, visit stemcell.com/immunocolt-XF.
Now we are at the second part of our episode today, and we are very happy to have with us Professor Anna Anderson. She is Albert H. Coons Professor of Neurology at Harvard Medical School. And the Anderson Lab studies a lot about mechanisms regulating T-cell responses in cancer, in chronic disease, and also the role of inhibitory and immune checkpoint inhibition in treatment of cancer and in the function of T cells and regulatory T cells. So I'm very happy to have her. Thank you so much for joining us, Professor Anderson, to the Immunology Podcast.
It's my pleasure to be here. Thank you for the invitation.
Pleasure to have you. All right, Brenda, it's T cells. You have
T
to
cells.
go first.
It's the best cells. We have this joke that Jason likes enterocytes, and I like the proper immune cells, the ones T cells he's not in use. So
T cells are also important in the gut. So
they are. Yeah.
See?
If this was innate lymphocytes or memory resident T cells that we're really driving into, I'd shove her off the front of the line immediately.
Well, so because we know T cells are the best cells, let's talk a little bit about our current understanding of T cells and in cancer therapy, we already have many guests, looking into T cell responses. But I think, Anna, you are a great guest to talk about the importance of co-inhibitory molecules and how our understanding of the elements that regulate T cell responses is currently, and how do we use that to treat certain diseases such as cancer. Maybe you want to give us a quick kind of update for us and our listeners.
Sure. So we've seen obviously a lot of success with PD-1 checkpoint, CTLA-4 checkpoint, LAG-3 now approved in combination with PD-1. But I think our understanding of the differentiation across these molecules, because there are increasingly more molecules getting the label of checkpoint receptor. And I think our understanding of what the nuances are. Not all these molecules do exactly the same thing. They may not necessarily be operative in the same tissues to the same extent. And the ligand receptor relationships get even more complicated as we go further down the sort of list of checkpoint molecules that we've been studying. So I think understanding the biology of these molecules will help us to be more informed in approaching how we target them and capitalize on their biology for therapeutic benefit for patients.
So we had this big success. I'd say early. I mean, it took a lot of work to get there, but like once it got in the clinic, you kind of had this runaway early wins. And then it seems to be a lot of very incremental fighting for the next inch of turf, right? Like
Absolutely.
very hard. What do you think is causing that? Is it there's more to the checkpoint story? Is it, I mean, and there may not be one thing. Is it blockades insufficient? Is it the drug doesn't get to the spot you need it to in a lot of these cancers? Like, where do you think the root, like, hey, we ran into a wall of the checkpoint story more recently what is that wall?
So to give you, I think it's a little bit of each of those things, but I like to think of the checkpoints in a bit of a hierarchy. And at the top of that hierarchy is CTLA-4. And we know this coming from the autoimmune field or study of self-tolerance. If you are deficient in CTLA-4, you will have a lot of issues with inflammation. And you could develop a lot of autoreactivity. With PD-1, you can also see that. We've learned that from studies and experimental models. If you lose PD-1, you can get some loss of self-tolerance. You don't really see that with the sort of the next generation of checkpoints like the TIGITs, TIM3s, LAG3s. So there's that. I also think really what the data are telling us, what we've learned from all these clinical trials of combining, PD-1 with everything under the sun, is that we need to really remodel the tumor microenvironment because T cells can only do so much when they're in such a nasty, unfriendly tissue environment. So we need to change something else. I think focusing exclusively on T cells is just not going to be enough.
So not only looking into those receptors that are indeed expressed on T cells, but maybe also other receptors that influence the function of other immune cells. Are we talking myeloid cells? Are we talking innate? What should we be looking at?
I think the myeloid compartment is very dominant in many cancers. For example, glioblastoma has more myeloid cells than T cells. So I think focusing on approaches that could remodel the myeloid compartment. But I also think the stromal compartment, the fibroblasts, are increasingly being investigated for their properties. And it's not just one type of fibroblast. There are all these subsets now of cancer-associated fibroblasts. And many tumors are heavily fibroblast rich. And those are generally the ones that are hardest to treat.
Yeah. So I would like to, maybe we could get into a bit of what does it mean to try to bring some of these discoveries that we do to the clinic, and a bit what are the challenges that become evident when Uh, for example, you mentioned TIM3, and you have done a lot of work on looking at TIM3 as a potential molecule to inhibit for the purpose of improving the immune response. And I would like you to maybe share with us a bit what has been. So TIM3 recently has gone into clinical trials, although it seemed very promising, and additional checkpoint inhibition in mouse models and in preclinical studies. The actual clinical studies were not as satisfactory as expected, and I think this is a really good example of the mouse to human barrier that we see, and also um, a good opportunity to reconsider how we understand checkpoint inhibition, or what exactly is that we're doing to these cells. Maybe given that you have such a good experience with this, would you maybe share with us how the story goes, and what do we learn from this kind of results?
So the important thing about TIM3 is that it's expressed on terminally differentiated T cells.
Mm-hmm.
So number one, I think the initial thinking around TIM3 was that targeting it by blocking it on T cells was really the biology that we were looking for, and to
Right.
capitalize that in therapy. But what we've learned throughout the years of continuing to study TIM3 is that it has a very important role in dendritic cells. And its role in dendritic cells is controlling its sort of tolerogenic versus inflammatory state. And that's really important during priming or the initiation of T cell responses. And so, what I think we may have missed the mark on how to give TIM3 therapy in a way that would really benefit a patient. And what's happening actually now is the immune checkpoint field is now looking at what's called neoadjuvant therapy, where they give the checkpoint before surgical resection of the tumor. And this is being done primarily in melanoma tumors. And there, the initial data looking at PD-1 alone versus PD-1 plus TIM3, and this is publicly presented data, so it's okay to share and discuss it, is that having the TIM3 present doubles the rate of response in that setting. And I believe it's because we're capturing this activity on dendritic cells that is there to really prime and initiate T cell responses, which is basically the concept behind neoadjuvant therapy. It's kind of a terrible nomenclature for scientists to think of. Neoadjuvant is, the terminology is a little funny, but that's what it is giving therapy before surgical resection. So I think that's one of the reasons underlying sort of some of the poor responses that have been seen in clinical trials. The other biology is the heme malignancy, or myeloid dysplastic syndrome. So that's the other place where TIM3 has been investigated in clinical trials. And the early data was fantastic, but there were very important differences between the early trials and the later trials. So the phase 2 trials had a different chemotherapy combination. So it was decitabine versus azacitidine, which was what was used in the phase 3 trial. And the types of patients were different. So in the phase 3 trial that didn't meet endpoint, they had selected for high risk myeloid dysplastic syndrome patients as opposed to intermediate risk, which was in the earlier trial. So you had a sicker patient population, you changed the chemotherapy, and now, we'll never know, because I don't think anyone, at least at this point, is looking to redo that same trial with a different patient population and a different drug combo, because it's a big investment to do that.
I find it so interesting. Yeah, the nuance. It's so hard when I remember reading about the results of the trials and thinking, oh, that's a shame. Oh, what did we miss? And there's so much nuance when not only the science itself, but also the actual execution of this trial. Sometimes some things are beyond the control of the researchers, or sometimes some of the way that you explain it makes me, that's not the way I had seen the results of these trials. It's so much more complex, that there's a lot of complexity that is lost in this kind of one-liner, biotech headlines. They're like, oh, Novartis is dropping their TIM-3 therapy, and people were so wrong. And it's very interesting to look at this with nuance. I think that's in the end that's
Yeah,
always lost.
it's like we're learning in real time from the patient data. And it also speaks to when you can collect samples from patients on trials, you really learn as to what's happening. If you just sort of read out clinical endpoints, sometimes you're left with a lot of unanswered questions.
Yeah.
So what's next for TIM-3?
I mean, I would love to see how this neoadjuvant trials pan out. I'd love to see someone combine it with a vaccination therapy. I think that's probably going to be the best scenario based on the current understanding of the biology of that molecule.
So there are neoadjuvant trials with it going on, so it's not completely shelved.
Yes. So what I quoted to you is an actual trial being run at the University of Pittsburgh Medical Center, comparing PD-1 versus PD-1 TIM-3 in the neoadjuvant setting. And the data's really quite good.
So another thing that sometimes I think we don't pay enough attention to is when trying to understand, in checkpoint inhibition, or the role of receptors that get upregulated in certain populations of T cells. And it's something that I discuss a lot when students are presenting in the lab, is what exactly are these proteins? So we know that, for example, PD-1 will also has some phosphatase activity associated, and it will really directly affect the signaling within the T cell. Maybe CTLA-4 has more to do with blocking the co-stimulation of CD28, because it binds to the ligands of CD28 with much higher affinity. So what does TIM-3, for example, or other of the other potential checkpoint inhibitor targets that we talk about, do they have intrinsic signaling function? Are they just binding to what do we know about what happens inside? They're not just activation markers. They're more than that, right? They have a function biologically.
Yeah, absolutely. And that's a great question. So perhaps the one we know most about is TIGIT. TIGIT actually has a similar inhibitory signaling motif in its cytoplasmic tail. It's called an ITSM. TIGIT also it's kind of like a hybrid. It has an inhibitory signaling motif in its tail, but it also is part of this family that has a co-stimulatory counterpart, which is CD226 or DNAM1. And they have different affinities, similar to like CTLA-4 competing with CD28 for B7. So TIGIT can compete with DNAM1 for binding to CD155 or PVR. So it gets multiple mechanisms kind of layered on top of each other. LAG3 is one that more recently the biochemistry was finally worked out, how it might work inside cells to inhibit T cells. And it has this kind of really unique mechanism. And this is work done by one of my colleagues and friends, Dario Vignali at University of Pittsburgh. And it has these motifs in the tail that acidify the local microenvironment around where the molecules are concentrated, and that affects signaling proteins. So it's not your sort of traditional inhibitory signaling motif that you might find in other co-inhibitory receptors.
So is there anything new on the horizon you can talk about in terms of, okay, well, we have TIM-3, it's in clinic. So, from a PhD perspective, right, that's boring. It's in the clinic. But is there anything new that's next that you want or you're looking at that you hope gets in the clinic?
Ooh, in terms of checkpoint, I think we're kind of saturated,
Exhausted
really.
even.
Yeah, exhausted to use a pun. I think for what I'm interested in is other ways of altering T cell states. And although I'm cautious around over engineering T cells, I think there's a lot of potential around the adoptive cell therapy field right now. And there's certainly that's where you see a lot more of the excitement is in CAR and every iteration of CAR, whether it's engineered TIL or TCRTs, CAR Ts, modifying those cells to do the best job in vivo. I think that's, and I think CARs for autoimmunity is really exciting area of growing research right now.
Yeah, for sure. I also find that super interesting.
Yeah. My background is autoimmunity, actually. That's where I started my research career. So it's kind of near and dear to my heart.
Do you think the CARs can sustain themselves for that? So, when you think about CAR and cancer, right? This is very simple, right? But like you put a CAR and it kills the cancer. I know there's some cancer that can hang out, but like the idea is it kills it all, and then it can go into retirement, right?
Yeah.
But for autoimmunity, can this CARs go into retirement, like it finds the autoreactive cells and gets rid of them all and then goes into retirement? Or is it something where you're stuck with a sustained mode because you can't unprogram you can suppress the autoimmune program, but you can't get rid of all the parts that drive it with a CAR?
So time will tell, but what's interesting is, and I've heard this argument made by the people who are studying autoimmune CAR T, is the burden of disease and cancer is so much bigger than what you have in an autoimmune scenario that you don't even need the amount of CAR T cells that you would need for cancer in an autoimmune setting. You can give, essentially they're looking at giving just like one bolus of cells and you're done. And it's not even, it's probably, it's much lower magnitude of what you even need to deliver in the CAR product to achieve results. So I think that's very interesting, but how long lasting that response will be is something that we'll only be able to know with time.
Yeah, I guess people haven't been treated for long enough. But yeah, but a part of me really thinks that if you really wipe out all of the B cells, all of these reactive B cells, then you kind of get to start over. And maybe the same, if you don't get exposed to the same triggering mechanisms that initiated the autoimmune response in the first time, maybe you can get away with it.
Yeah, it's also a trade-off, being on immunosuppressive therapy for a long time versus, if you get some CAR Ts once every 5 years or something, that might be
Yeah.
preferable to being perpetually immunosuppressed with anti-TNF or anti-IL-6 or what have you.
Yeah, for sure. So in your opinion, what is like your podium, your top three um things that we need to think about for CAR T cell therapy, for cellular therapies to progress in the clinic, to probably fill the gaps that other therapies cannot fill? What do you think are your preferred topics of research in this sense?
I think it's going to be control, meaning that how do we control those cells? Because it's not without some significant fallout sometimes in treated patients where we get cytokine release syndrome, and so how do we kind of get just the right mix of cells? And CAR is not just one homogeneous population of cells; it's often a mix of CD8s and CD4s, and they're at various states. So how do we get the right combination of CD4 help for the CD8 cells and also uh, what states of T cells, especially we want cells that will persist in tissue. Ultimately, if you could get a car that adopts sort of a tissue residency memory type program, that would then provide sort of some sentinels that'll hang out in tissue in case there's a recurrence, and then just, zap it once it manifests. That would be, I think, really kind of interesting.
Yeah, do we know if any of the cars now in cancer, other cases, switch to a resident phenotype on their own just by once they get back into a human and hang out there?
Ooh, that I don't know, because I'm not very active in research in the CAR T
Yeah, I
cell
didn't.
field, but I do know that the presence of cells that have markers of stemness in the infusion product has been correlated with better response, uh, sort of long term. But especially for solid tumors, I think that's going to be what's important, because the problem with solid tumors is you get recurrence. I mean, there was some like great kind of anecdotal reports of glioblastoma with CAR T cell and showing an effect, but then eventually all the patients recurred. So that's the real challenge for solid tumor CAR T therapy.
It's good that you mentioned the concept of stemness, because I just want to connect it with some of your recent work
Mhm.
on the role of the transcription factor TCF1, also known AKA TCF7,
That's
the
the
protein.
gene, I know. Not my fault. Sorry.
I found that is so confusing that
I know it
the
is.
genes of, in case some of our listeners are not aware of this, the gene and the protein are called different numbers. Well, so, and it's good that you mentioned that, because that is also something that recently, the idea of stemness, no, the stem cell, the self-renewing T cell phenotype that we want. Um, Sometimes I feel like it's one of those things that are said a lot, and it's not clear what they mean. And people desperately want this to mean something very important,
Yeah.
and the point doesn't always get across. So maybe let's talk a bit about TCF1 stemness and the role in T cell biology from the perspective of what your lab has been doing and your personal
Mhm.
research on that.
Oh, thank you for that. I love that question. So, stemness is a term that gets thrown around very loosely. And the ultimate stem cell is, in terms of the T cell compartment, is your naive cell. And naive cells are very high for TCF1, encoded by TCF7. And what we refer to in the field as stem-like T cells are T cells that have seen antigen. They have turned on PD-1 expression, but it's low levels compared to what we would see on exhausted or dysfunctional T cells. And because of that, sometimes they've been referred to as progenitor exhausted T cells, or TPEX, which I personally think is an absolutely terrible nomenclature, because the cells are not exhausted. They're not
Yeah.
progenitors. Progenitors are cells that can give rise to multiple lineages of cells. Those are the cells in your bone marrow, for example. But so I think of them as stem-like precursor cells. So they are cells that have seen antigen, so they have the specificity for, in our case, a tumor antigen. And they have still some TCF expression, so it's a little lower than naive cells, but it's higher than a full-blown effector cell. And because of that, they are able to persist. And so they retain, so-called stemness memory programs. What we've done in my lab is really try to understand, well, what is TCF doing? And the one answer that we've come up with so far is that TCF1 presence helps poise cells for response to low T cell receptor triggering. So if you have suboptimal antigen, low levels of MHC, perhaps a low affinity antigen, you need those cells to kind of hang around for a long enough period of time to get going, get triggered, and then engage an effector program and expand and do their job. But I think there's still a lot of unanswered questions in terms of biologically, what are the mechanisms by which TCF really does this in T cells?
So I guess what's next on what you're going after that you can share that or postdocs available or grad student slots, the usual plug of, hey, I'm really getting into this now. Come hang out in my lab if possible. I know it's
Right.
weird times in Scienceville right now, so I'll put that asterisk out there ahead of even you putting it out there.
Yeah. So two things that we're really excited about is we've done a lot of analysis of T cell states using all the omics technologies and such. And one of the things that I'm most excited about is understanding how perturbations that happen in early T cell states imprint the trajectory of what those cells ultimately become. And I think once we really understand these complex networks of how things can be programmed from early on, it'll give us a lot of knowledge and a lot of power to then manipulate T cell functions. The other thing that I'm really, really excited about is the interface of immunology, neuroscience, and cancer. So we are actively studying pathways by which T cells have essentially borrowed a pathway that normally operates in neurons to regulate their responses in cancer. And that really has me excited because it's a whole new area of research.
Is that associated to your work on glucocorticoid signaling? But that's already some time ago. Or what
No,
part?
we have work that's in revision right now where we're
Mm.
looking at pain sensing and interference with nociception and
Mm.
how this operates in T cells to disable their responses in settings of chronic stimulation, like in cancer.
Yeah, it's established that chronic opioid use and other things are immunosuppressive.
Yeah, but the mechanisms are very poorly understood
Yeah.
and they're kind of sort of, loose
Something beta
connections.
arrestin, right? And that's about it from the GPCR coupling, and that's all they've ever figured out. But it's like clinically, it's established that there's some pretty good data that immunosuppression or chronic pain becomes an immunosuppressive.
Right. And we all know that inflammation is part of pain, right? It's one of the cardinal signs of pain. Yeah. So we know that there's a connection between, immune activation, driving inflammation causes pain, and then the mechanisms that interfere with pain, we think are actually also directly interfering or suppressing immune activation. So it's kind of all interconnected in a circuit.
All right. I think that we could talk about. Well, want to wait for your paper so we can cover them in the podcast when they come out. So let us know. Uh, we like to conclude our conversation asking our guests a question to get them to know them better, or for our audience to get them to know better. And I like this one, which is what is one hobby you've always wanted to pursue, but because you're, in a busy science life, you've never had a chance to, or maybe you do and you just want to share with us. Either one is fine.
So the one thing I've always wanted to do, and I think I just need to start doing it before I get older and have less time, is rowing, actually. I There's something that attracts me about being out on the water, and I've never actually done this, and I should say I'm pretty terrible at canoeing. I did that once, I was horrible at it, but I love the sport. I love the head of the Charles Regatta is one of the highlights of the fall season in Boston. It's a worldwide regatta, and I love going there, watching all the races. And my son rode for a year in high school, and I thought it was like I was so happy. I thought I was more excited about him doing it than he was. So I think I'm just going to have to learn and try it and do it myself.
But is it the rowing, or you want to be the person in the
Well,
front, just telling everybody else?
oh no, I want to be doing the actual rowing, not the coxswain. Yeah.
Coxman, yeah.
Yelling at people through the
Row
bullhorn. Yeah.
faster. That's what they do, right? No, just kidding.
Yes. Go, go, go.
That sounds fun. Well, I also love water. I've recently started learning to sail. And I think that's
Oh, that's
also,
exciting.
yeah. So that's maybe not as physically demanding as a rowing, but definitely being on the water has
Yeah.
a certain
Appeal.
Yeah.
There's something about the fresh air, the water. Yeah.
Yeah.
Hard to beat that.
Well, maybe some Cambridge, Massachusetts team that needs a new member can contact Anna and invite her to join their team, so you can find the motivation to get on it, to get on with it. All right. So thank you so much. We've had Anna Anderson from Harvard Medical School joining us at the podcast today. Thank you very much for joining.
Oh, it's my pleasure. Thank you for inviting me.
Thank you for coming on.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including episode summaries and links to all of the interview and Roundup papers. You can also reach out to us on X at Immunopodcast, Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com with feedback or if you want to suggest a guest. See you next time.